Welcome to Infection and Immunity Evidence Explained, a Doherty Institute podcast. I'm Rebecca Elliott. Our immune system is made up of many types of cells. Many people will have heard of T cells that fight infection, but did you know there are many different subsets of T cells? Well, today we're going to talk specifically about tissue-resident memory T cells and a new study just published in the prestigious journal Science by Doherty Institute researchers. The team found new ways to remove immune cells that cause skin autoimmune diseases without affecting protective cells that fight infection and cancer. Here to tell us about this latest research is Dr. Simone Park. Simone is an immunologist who aims to understand how the immune system responds to infection and cancer. She completed her PhD at the Doherty Institute in 2019 under the supervision of two authorities in memory T cell biology, Professors Thomas Ghebhardt and Laura Mackay, and holds an honorary appointment here. She currently resides in the US, where she is a Cancer Research Institute postdoctoral fellow at the University of Pennsylvania in Philadelphia. Simone, thanks so much for joining me today on Infection and Immunity Evidence Explained.
Thank you for having me.
So first and foremost, can you tell me what is a tissue-resident memory T cell?
Yeah, so T cells are what we might think of as cells that would be found in the blood, because a lot of the T cells that people might be familiar with are those ones that are found in the blood and that are constantly migrating through the body. But tissue-resident memory T cells are a bit different to those cells. These cells are actually found in the tissues of the body, like the skin, the gut, the lung. They're found at the barriers where infections and microbes might actually be able to enter the body. And so these are specialized types of T cells that are really important to protect us against these microbes that might enter through these barrier surfaces like the skin and the gut and stop these microbes from being able to get any further into the body as quickly as they can.
Why are they important?
Yeah, so they are very important to fight infection, but in the past few years, we've started to realize that they're actually also very important for maintaining tissue homeostasis as well. And this encompasses both protection against the progression of cancer, so mutating cells, controlling those cells so that they don't become fully fledged cancer cells that can develop into nasty tumors and progressively grow, as well as maintaining tissue repair. So when our tissues get injured, so for example, if you get a cut in the skin, these cells can also help to accelerate the rate of tissue repair and help our skin or our barrier tissues to repair itself more quickly.
All right, so we know these cells can fight infection and cancer, but they can also cause autoimmune conditions. Why and how does this happen?
Yeah, so as well as us understanding more about how these cells are protective, like you just said, we've also started to understand that these cells can actually turn bad, I suppose, and start to cause potential autoimmune disorders or autoimmune symptoms. And this typically happens when these cells might recognize parts of the body that should be ignored by the immune system. And these are parts of the body that would be thought of as self, or your immune system usually recognizes these parts of your body as things that shouldn't be attacked. But sometimes that message, the wires get crossed and the cells can't really understand that they're not supposed to attack these normal tissues of the body. And so just like any other type of adaptive immune cell, tissue-resident T cells can also be directed against these self proteins in a way that they shouldn't necessarily be. And this is what would lead to autoimmunity.
And that goes back to Peter Doherty's, why he won the Nobel Prize.
Yes, yeah, understanding antigen recognition, exactly.
So how are these autoimmune conditions currently treated?
So these autoimmune conditions, normally we need to treat the symptoms rather than the cause of these conditions. And so a lot of the drugs that we currently have on offer in the clinic, ones that are going to stop the kind of effects of what the immune cells are going to start to do. So it'll dampen their responses so that they're not able to attack the body as easily anymore. Or if they are attacking the body, it'll stop the tissues from being damaged as much. But these don't actually remove the cells that are causing the issue. And so this means that these treatments, we need to keep giving them to patients for really long periods of time, potentially for the life of the patient, because we're not actually removing the cells, we're just stopping the cells from executing their functions.
Well, let's get to your discovery. So in your most recent work published in Science together with Professor Laura Mackay and Dr. Suzanne Christo, you found that in an animal model, you were able to distinguish between the disease-causing and the disease-fighting immune cells. Can you tell me a little bit more about your findings?
Yeah, sure. So we know now, as we've just been talking about, that there's many different types of T cells, but we didn't really know before this study if those types of T cells, these tissue resident cells that cause disease versus cause autoimmunity in different contexts, whether those cells depend on different things. And this is something that it was really important to find out, because we need to know if those different types of cells that are causing different sorts of issues in the tissue or potentially being protective rely on different mechanisms to survive within the tissue. If we're going to design drugs, that we might be able to just remove the cells that are causing problems without removing the cells that we've discussed are important for protection against infection, cancer, and for promoting tissue repair. And so, yeah, our findings basically showed us that those cells do require different things. And by virtue of the fact that they require different things, or they use different mechanisms to survive in the tissue, this means that we can then target these differences between them to potentially develop new drugs in the future. And in our study, we basically showed that it was possible to target these two different sets of mechanisms found in these different types of T cells and remove just the bad cells without removing the good ones in animal models.
Well, congratulations. That's just truly amazing work. You must be wrapped with it.
Yeah, it's been a great study to be a part of, but it's also been one that's taken us a really long time to publication, especially because I've now moved overseas. So it's been really exciting for us to finally be able to share the results and tell the world what we found, because we've not been able to do that on a broader stage until very recently.
How could this potentially change the way that autoimmune conditions are treated?
So hopefully in the future, if we can use this information to develop new drugs that target these differences between the different types of immune cells directly, then we might be able to use those drugs to remove the cells that cause autoimmune disease and that are constantly causing these symptoms in patients permanently. And so that would mean that potentially we'd be able to generate drugs where we wouldn't need to continuously give them to patients. We would just be able to potentially give them to patients for a short time, remove the cells, and then they could actually have more long-lasting effects of that treatment than what's currently available. So that's the hope. And in addition to that, a lot of autoimmune disorders, we don't really know exactly what pathways to target mechanistically to get rid of the cells that are causing the problem. And so hopefully by shedding light on the different mechanisms that are used by different types of cells, we're making sure that we can actually get rid of the right cells. And that would mean that hopefully we can generate drugs that will be more beneficial in a wider net or population of patients than what potentially might be currently available.
We talked about the fact that this research was done in an animal model. So how long would it potentially be before we might be able to see your work translate into humans?
Yeah, it's a great question, $1 million question. So our research very much focuses on understanding the mechanisms of how these cells work at a very fundamental level and unlocking the sorts of molecular pathways that are going on inside the cell. And so our focus in our lab is not so much to translate this information into therapeutics, but we're hopeful that pharmaceutical companies, other people that are working more in that therapeutic space are going to pick up on this information and start taking it to the next level to be able to start developing new targets. And there has been hopefully there's going to continue to be interest in doing that now that we've published our findings. So hopefully soon, if the targets that we've found might also prove promising in human clinical trials.
What are the next steps for this research?
So in terms of what we're doing at the moment to expand upon this research, we're doing some more sort of validation or expanding our understanding of how these cells might differ between mice and humans, mice being the animal model that we used. So we have been able to show that there's conserved use of different pathways that we discovered in our study in humans as well. But we've been following this up specifically in patients that are suffering with disorders like psoriasis, alopecia, and vitiligo as well, to understand more about how the landscape of the immune cells in those diseased tissues compares to animal models, but also to healthy subjects. And this should also help us understand how this information could best be translated in the future.
Fantastic. So this is not the first time that your research has made a prestigious journal and headlines. In early 2019, you published research in Nature, which described tissue-resident memory T cells, so this same subset of T cells we're talking about today, can control melanoma. Can you tell me a little bit more about that work?
Yeah, sure. So that work was done during my PhD at the Doherty Institute. And in that study, we discovered that tissue-resident memory T cells are really important to protect against the development of melanoma. And they can do that by entering the skin and basically controlling melanoma cells in a state of sleep or a dormant state. So they stop the melanoma cells from growing, but they don't necessarily completely kill or eliminate the melanoma cells. And there's sort of a ongoing communication or crosstalk between the melanoma cells and the tissue-resident memory T cells that allows the tissue-resident memory T cells to control the melanoma, even though the melanoma cells are technically still there. And so that was the first time that it had been shown that tissue-resident memory T cells have this sort of protective role in control of melanoma in the skin, because the field had sort of recognized that these cells associate very well with protective responses against cancers in clinical trials, especially immunotherapy trials, but it wasn't really clear why those cells might be so beneficial in patients in these trials and in these settings. So this really shed light in understanding that these cells can protect against cancer development and progression, and how they might be potentially able to do that.
So this work led you to receive many grants and awards and a Cancer Research Institute fellowship at the University of Pennsylvania, where you are now. So tell me about your role in the US.
Yeah, so now I'm working at the University of Pennsylvania in Philadelphia with John Werry, and he is a leading researcher in understanding T cell dysfunction. So this is a process where T cells can stop being able to execute protective functions where they can protect against infection and cancer, because they lose their ability to do so through a series of events that occurs within the cell. And so my work is focusing on understanding how that process happens at a very granular level inside the cell, how DNA changes when this happens, how proteins are regulating this process, and whether we can then use that information to understand how we might be able to develop better drugs and immunotherapies to reinvigorate these exhausted or dysfunctional T cells so that they're better able to fight chronic infections and cancers. And specifically, I'm looking mostly in tissues again, because that's what I love. I love looking at T cells in tissues and understanding how these tissues play a role in influencing the functional abilities of these cells and in influencing the states of these cells, and how we might be able to use that information to really help improve these cells' functions in tissues and tumours in patients.
It sounds like absolutely fascinating and necessary work, so we wish you all the best. But will we see you back in Australia?
It's a great question. I think that it probably depends a lot on the research funding environment. Yep. But I mean, yeah, it's possible. So yeah, probably up more so to the powers that be, but I wouldn't rule it out. I love Australia. I would love to come back one day. So we'll see.
Simone, thank you so much for joining me today.
Thank you so much for having me. It's been great to talk to you about this work, and also just to be on the podcast. And it's great to, stay connected to the Doherty.
And thanks to all of you for listening to Infection and Immunity Evidence Explained, a Doherty Institute podcast. If there is a specific topic you would like us to cover, please get in touch with us by emailing doherty-media@unimelb.edu.au. That's doherty-media@unimelb.edu.au. And if you've enjoyed our podcast, please rate and review on your favourite podcasting app and subscribe if you haven't already. The Doherty Institute is a joint venture between the University of Melbourne and the Royal Melbourne Hospital. See you next time.